So this is the topic of gene technology, and in this topic we're going to explain how recombinant DNA is produced, including the role of restriction endonucleases and DNA ligase. Now, as you know, the genetic code is universal. It works the same in all organisms. Therefore, it is possible to swap genes between different species. This is called genetic modification or genetic engineering, and is being done on plants and animals for all sorts of uses. In this topic, you will learn how to isolate a gene and transfer it into another organism, forming a transgenic organism. Now, in order to carry out genetic modification, scientists need to do various processes step by step and use specific tools. They need to isolate the gene that they want to transfer. They need to then insert that gene into a vector and then use that vector to transform the host's genome by inserting the recombinant DNA into the host. DNA from one species, which is then being combined with another organism's DNA, is known as recombinant DNA. The first step in creating this recombinant DNA is to isolate the desired gene from the genome of the gene donor. Now, there are two main methods we'll look at for this. One is the use of restriction endonucleases, and the other is the use of messenger RNA and reverse transcriptase. Restriction endonucleases are made by bacterial cells as a defense mechanism against viruses. What they do is they use these enzymes to cut up the virus's DNA into fragments. Therefore, what we have done is to isolate this enzyme and use it to cut up the DNA that we want into sections. What they do is they target specific sequences of about 4 to 8 nucleotides long called restriction sites. So different restriction enzymes cut at different restriction sites. And there are many different restriction enzymes found in different species of bacteria. When they cut at these different restriction sites, they usually leave short overhangs of unpaired bases called sticky ends. Now, these join very easily to complementary sticky ends. So, if you use the same restriction endonuclease to isolate the desired gene and the same one to open the host genome, then they should complement very well, and it'll be easy to stick the desired gene into the host. However, one potential issue with restriction enzymes is that when you cut the DNA that you want, it will cut anywhere where that restriction site is, even if it's in the middle of the gene. So it can be really good if you can get restriction endonucleases that will cut either side of the gene to create the right restriction fragments, but it can be not perfect. So in that case, you may want to use the other method, which is the use of mRNA and reverse transcriptase. What's reverse transcriptase? Well, do you remember the process of transcription in protein synthesis? This is when the DNA code for a gene is used as a template to make single-stranded messenger RNA. Well, what reverse transcriptase does is it does that process backwards. It takes the messenger RNA and turns it back into the DNA. We get it from a retrovirus. We isolate the reverse transcriptase from a retrovirus, and these viruses have an RNA genome. So they use this enzyme to turn their RNA into DNA, so they can then use that DNA to infect their host. So what we need to do is find a cell where the messenger RNA that we want is being actively expressed, and so there's plenty of messenger RNA for that gene. You take that mature messenger RNA, which has been spliced to have its introns removed already, so that's a benefit here, and we use reverse transcriptase to make the complementary DNA. That's only going to be single-stranded, so the final step is to use DNA polymerase to make it double-stranded, resulting in a copy of the gene called cDNA. Now, there are some advantages of using reverse transcriptase and messenger RNA over other methods. Firstly, it's easier to get the complete gene than from the whole genome. Secondly, it's available in greater quantities because of all the messenger RNA. Thirdly, prokaryotic cells cannot remove introns from a gene, and in genetic engineering you quite often use bacterial host cells. So if you insert the DNA with the introns, it's a problem, but in this case the introns have already been removed, so that's another benefit. Once you've successfully isolated the gene, you now need to insert it into a vector's DNA that will then take it into the host for you, and to do that we use enzymes called DNA ligase enzymes. These form covalent phosphodiester bonds so that you can join, as is shown by this diagram, the sticky ends together.